Right, today we're going to take a look at enzymes. This is the first section in our cellular energy dynamics unit that we're going to get to here in Unit 3. So we are talking about enzymes. And when you think back to Unit 1, where we had discussed proteins, we had previewed that enzymes were going to be a portion of that umbrella that proteins covers. So we'll start with just that idea that enzymes are proteins that speed up reactions. And when they do this, they are not consumed. So they're proteins that speed up reactions without being consumed. So in biology and in chemistry, we call things that do that catalysts. They are biological catalysts. So an example that potentially we're going to be able to use in the lab would be catalase. And catalase you can find in liver of any animal tissue or most animal tissue. And then another one would be peroxidase. And peroxidase you're going to find in plants. And in Unit 2 we talked about the organelle that's responsible for housing these materials, right? It was the peroxisome. If we go back and look at our kind of cell diagram here, this peroxisome is where you would find this catalase and this peroxidase. And again, the role is to break down hydrogen peroxide. So this is what it looks like. So cells, they generate hydrogen peroxide, which is 2H2O2, right? They generate this as part of reactions, and that needs to be dealt with. Otherwise, it's going to be maladaptive and potentially dangerous to the cell. So we call this the unit that we are going to introduce the catalyst to get it to break down or to be built is called the substrate. So we start with what we call the substrate. And in this case, the substrate is hydrogen peroxide. We introduce the enzyme. The enzyme is not consumed or used up, so we put that typically above the arrow. So that's peroxidase. This is our reaction arrow. And that's going to decompose this peroxidate or this hydrogen peroxide into water. And to keep it balanced, we'll put a 2 there. So 2 molecules of water plus oxygen gas, right? And these are what we call our products. So we have our substrate, we have our enzyme, and we have our products. Okay, and again, the role of this enzyme is to deconstruct this substrate into the products. Or this could go the other way. If there was an enzyme that actually built, this hydrogen peroxide from water and oxygen gas, that would also be something that can occur. These enzymes are very, very, very specific. So they are shape specific. So we'll say that enzymes, they change shape of the substrate. This is how they function into an unstable one. So they change the shape from a more stable structure to an unstable shape that has more potential to break apart, or in the other manner, be built. So it changes the shape of a substrate into an unstable one, or increases the opportunity to be built or to bond. So an example that I like to use is an example with just a key ring. Right, so again, if this is my substrate, this ring of keys, and if I try to pull these keys off this ring, this would take an extremely long time. Eventually the metal might get weak and eventually the keys might come off, but that would take a really long time. So I like to think of an enzyme as being your hands, and the ability of this enzyme to change the shape of the ring in a way where this reaction is more likely to occur. So by changing the shape of my substrate, the reaction, in this case it's just keys coming off the ring, can happen. So again, what's happening is this enzyme is changing the shape into an unstable one where it's more likely that this reaction might happen. And again, a key ring is a example I like to use because it's easy to remember. So again, we'll go ahead and we're going to sketch out just what this looks like. So here we've got, this is peroxidase. We'll say this is peroxidase. And this is the enzyme. And our substrate is going to be hydrogen peroxide. So we'll say this is H2O2. So this is our substrate. And the site at which the substrate binds to the enzyme, we call that the active site. So we're going to get the substrate binding to the enzyme. We're going to see a change in shape. This peroxidase is going to change its shape, and it's going to put this hydrogen peroxide into an unstable shape. And what's going to result will be the release of oxygen gas, which is not going to be dangerous for the cell to deal with, and water. We call these the products. Again, we said a good example would be this keyring example, right? The keyring example is one that you want to try to remember if you can, because it gives you that analogy of that shape change. Right, it's making, again, we'll just put one other thing here. What we meant by this note above, it just makes this more likely to react, whether that's coming apart or being built. We'll just put that in there. Now, when we look at the reaction rate or a graph of a reaction, we can see the impact of an enzyme when we do that. So we're going to do that next. So I'm going to create a graph. And we're going to sketch this out. And I'm going to put energy over here. And I'm going to put time down at the bottom. So without my enzyme, I'm going to draw what this reaction looks like. Okay, so I've got here, this is my starting energy with my hydrogen peroxide. And if I want to, or maybe this is the energy of the total system that I'm looking at. Okay, so again, this is my H2O2, and this is the energy of my total system. If I want this to break apart, again, it would take a very long time for this to happen, for enough energy to come into this system for it to break apart. Eventually it will. And when that happens, we end up with our products: oxygen gas and water. The amount of energy that's needed to get this reaction to go, we can measure. So this amount of energy from where we are at kind of baseline up to the phase with which the reaction starts to go, we call this the activation energy. Activation energy, and we denote that E of A, or EA. And what we see is when we include an enzyme, so if we were now to redraw this reaction graph, if we include an enzyme, the amount of activation energy that needs to be put in the system to go is so much less. Again, because it's putting that product into, or that substrate into an unstable shape. So this reaction happens much faster, the activation energy is much less, and we get our reaction that proceeds much, much quicker, right? So this is with the addition of the enzyme. So bottom line, an enzyme is going to speed up your reaction because it lowers activation energy, right? That's the big takeaway, the aha from this, is that enzymes, they lower activation energy. So they lower activation energy. And by doing so, they speed up reactions. There's going to be some conditions that impact enzyme function. So we're going to take a look at those here. So we'll say conditions that impact enzyme function. So some different conditions that impact enzyme function. The first is going to be temperature. As we increase the number of collisions, this enzyme bumping into this substrate, we're going to see this go faster, right? So if we increase collisions, our rate goes up. But there is a limit. In fact, our range typically is from about 15 degrees Celsius to about 30 degrees Celsius. So that is our range within which our enzymes typically work biologically inside your cells. So there is a limit. If we get outside this range, typically that can make your protein unfold or denature your protein or enzyme. So that can denature, which means it's just unfolding. Typically it's that tertiary structure, the R group interactions that we talked about in Unit 1 can come apart. So outside the range it can denature the enzyme. So temperature is the first. Number 2 is pH. pH is the amount of hydrogen ions or these H3O molecules in a solution. So as you add substances to water that either freeze up hydrogen ions or you get these hydronium, these H3O molecules. And again, if you get outside the optimum range, which is typically going to be in the 5 to 7 zone, you get outside that range, again, those hydrogen ions start pulling at the side chains or the amino and the carboxyl groups of the individual amino acids, and you get that protein can denature, right? The enzyme can change, and when the shape changes, it no longer fits the substrate. We have some other things here that we'll take a look at. Number 3 is going to be concentration. Right, it makes sense that as you add more enzyme, you get more enzyme substrate interaction and you get a greater rate of reaction. Eventually though, if you're adding more substrate, that enzyme can only work as fast as it can work. And you can reach a saturation point in which you're just going to see a leveling off of rate reaction. So again, you can reach, we'll say, a saturation point with additional substrate, and there'd be no further increase in rate. If you just add more enzyme, you're going to see kind of an exponential curve on that, and you're always going to get a greater rate of reaction. Right? If you add more and more enzyme, there's just more working hands to pull apart that substrate. It doesn't work that way with substrate, right? If you add substrate, again, the working hands don't change, right? And they can be saturated to the point where you're just not going to see an increase. A couple other things that we'll just kind of make a note of, that there's something called a cofactor. So sometimes we have non-protein helpers that bind and aid activity. So these are called enzymes, cofactors. A lot of times these are going to be nucleotides. So these are non-protein helpers that bind and aid activity. So an example is going to be a cofactor that we see a lot here in Unit 3, and this is what we call NAD. NAD+, it stands for nicotinamide adenine dinucleotide, and it's a double nucleotide, a dinucleotide, that moves electrons in and around the mitochondria, right? So it's responsible for taking electrons to the mitochondria, dropping it off at the electron transport chain, and aiding in oxidative phosphorylation. We'll talk about all those themes and ideas later. Again, so this has a major role in respiration, cellular respiration. So we're going to see that later. Another one, we'll give you one more example, would be RAF. And RAF is going to be a cofactor that's utilized in photosynthesis. Likely it's going to probably be utilized by the enzyme called ribisco, which is something that we're going to see when we get to photosynthesis. So two different cofactors there, important for the function of an enzyme. Things that can inhibit an enzyme or make an enzyme non-operational, other than some of the things that we talked about above, would be called inhibitors. So inhibitors, they block reactions. And this can be done two different ways. They could be competitive, which means they are going to compete for the active site. So they are molecules that are shaped in a manner that will bind to the active site and not allow the substrate access. Competitive inhibitors, once those inhibitors break down, you know, if they have a half-life with how long they exist and they break down, this would be reversible, because once it's off the active site, then your substrate's going to bind. The other group is what we would call non-competitive. So non-competitive. They don't bind at the active site. But at another position which, so we'll talk about this here in just a bit more. So they don't bind at the active site. So if we pull back up our diagram of our enzyme, they're binding somewhere else, right? A competitive inhibitor is binding here, not allowing the substrate to bind. A competitive inhibitor, or a non-competitive inhibitor, is going to bind maybe down here, somewhere else, which is going to change the shape of the enzyme, and then the active site shape changes and doesn't allow access to that substrate. So it doesn't bind at the active site, but at another position, which can cause an enzyme shape change. Right, so when you talk about different toxins or poisons, sometimes that mechanism of action is going to be a non-competitive inhibitor to an enzyme, where it's changing the shape of an enzyme at a different position. That site, that position on which that binds, other than the active site, is called an allosteric position. So these are allosteric inhibitors. Right, so this means other site. So that's what we've got for you for enzymes. I hope that was helpful, and I'll see you next time.